Welcome to Verified RX, your prescription for success. Brought to you by the Vizient Center for Pharmacy Practice Excellence.
Handling cell and gene therapies is significantly more complex than traditional handling and sterile compounding due to their biologic origin, patient-specific nature, and regulatory oversight. Joining us today are two experts that are leading that conversation. Dr. Mark Wiencek, Principal Microbiologist, Technical Services Group at Contec, and Dr. Amanda Frick, Senior Clinical Manager, Market Intelligence, Pharmacy Solutions at Vizient. We'll cover compounding cell and gene therapies, risk assessments, and how to implement appropriate measures to avoid cross-contamination and occupational exposure in hospital pharmacies. I'm Carolyn Liptak, Pharmacy Executive Director in the Center for Pharmacy Practice Excellence at Vizient, and your program host. Mark, Amanda, welcome to the podcast.
Great to be here. Thank you so much.
And this is Amanda. Thank you so much for having us, Carolyn.
Yeah, thanks, Amanda, for being here. It's great to have you back. Mark, you're new to Verified RX, so tell us a little bit about your background.
Thanks, Caroline. I am a microbiologist by training. Got my PhD many years ago and been doing industrial microbiology since then, and all things from preservatives and cosmetics to water treatment to medical devices. And since joining the current company, Contec, I work on disinfectants and the microbiology of controlled spaces like clean rooms. But that has also led me to get interested and investigate and try to become knowledgeable about other things that can cause harm, like hazardous drugs, occupational exposure to drugs. And then, like today's subject, where these two things are coming together, where we've got microbes that could potentially be hazardous, but we've got to use them now as medicine.
Yeah, thank you for being here, Mark. We really appreciate your expertise. Let's get started just by talking about what types of cell and gene therapies are being utilized today.
When I hear this question, I think there are a couple of different ways you can think about it. One is how the therapies are being administered. We have cell-based gene therapies. An example of this is CAR T. That's probably the example that everyone is most familiar with. This is where the cells are genetically modified outside of the body. And then once the genetic modification takes place, these cells are given to the patient. This can be called cell-based therapy, ex vivo therapy. And we also have gene therapies where this vector carrying the genetic material is administered directly to the patient. This is also known as in vivo therapy. So we have these two different mechanisms of administration. That's one way you can think about it. You can also think about it in terms of how the genetic modifications are being made. At the DNA level, we can go in and edit the gene that's in place, and that is often achieved through the use of technology known as CRISPR, Cas9, and this can also be achieved, this genetic modification, through gene transfer. This is accomplished via a vector, and it's often a viral vector. And that's where a lot of this talk about hazardous handling comes in.
It's kind of an interesting thing about when does this become its own class of drugs. We may touch on this later of actually using other forms of microorganisms as immunotherapies, and one of those is BCG. That's kind of the old school cell and gene therapy before we got into all these molecular techniques of making these modifications. But as Amanda says, that's the big difference that as we go through this discussion today makes a difference of how does this come through the healthcare system and when do compounder pharmacies get involved with it. There's definitely some therapies where the T cells or stem cells are modified and then pulled from the patient, taken to a facility, could be on site, could be off site. They modify it, and then they bring that back to the patient, and they're essentially not compounded. And then the viral vectors is where the compounding pharmacies get involved.
That's really helpful to know. Thanks to both of you. Amanda, which one of these, or which ones, are most relevant to compounding pharmacies?
When it comes to which are most relevant to compounding pharmacies, I primarily think about those vector-based in vivo gene therapies. So the gene therapies that are being administered directly to a patient. This is where most of the compounding and preparation happens, and this is where pharmacists are working with the actual vectors as opposed to these cells that are being modified off-site. They've undergone testing to make sure they aren't infectious. The in vivo therapies are where these being most relevant to compounding pharmacies.
Yeah, the only other place that I've heard this is when they take these cells from the patient and take them to facility and they modify them, they are going to come back to that facility. And sometimes they're looking for the pharmacy to help with the infrastructure part of that. Like some of these things have to be frozen at minus 80 Celsius and just some assistance that I think they might come to the pharmacy. But ultimately, the viral vectors are what's coming as a classic drug in a vial, if you will, that now needs to be compounded and put into a unit dose for the patient.
In pharmacy, we use the NIOSH list as our gold standard for whether a product is hazardous or not. But what we're seeing is that many of these cell and gene therapies, the ones that you guys are talking about, are not necessarily on the NIOSH list. So, Mark, can you explain why they're not on the list?
Could have a whole nother podcast just about all the trials and tribulations of the NIOSH list, but it was decided to only have the new NIOSH list represent products that were reviewed under a part of the FDA called CDER, where the D in CDER is for drugs. The FDA has two different major arms of how they approve these types of drugs, and one of them is true drugs, and another section is basically biologics. It was just decided, not necessarily based on the hazard profiles, but more just the scope of what NIOSH could possibly cover. To traditional hazardous drugs, when you look at the kind of toxicological endpoints that triggered why we have to do special handling for these drugs, it was things about mutagenicity, carcinogenicity, teratogenicity, reproductive hazards. And those are really not the kind of hazards that we have associated with these biological drugs. It was just decided maybe there'll be a new list one day that gets into those kind of biologics, but it just was decided by the NIOSH group that they're only going to look at these traditional drugs that are approved through the drug part of the FDA.
That makes sense. But if these products are not on the NIOSH list, Amanda, can pharmacies consider them non hazardous just because they're not on the list?
No, it doesn't mean they're not hazardous. It just means that NIOSH doesn't have this formal guidance on them. In the absence of that direction from NIOSH, institutions are left to create their own policies for handling. And we're ultimately leaning on guidance from the CDC and NIH and how these viruses would be handled in a lab settings. In the lack of formal guidance, I think there is this big question in how we handle these things. That's why this is such a hot topic.
So you're saying they're not on the hazardous drug list, or they're not on the NIOSH hazardous drug list, but they are hazardous. And we know there are risks. Mark, can you tell us a little bit more about what these risks are?
Again, when we evaluate any of this, and this is kind of some of the NIOSH principles, you've got an inherent hazard, and almost anything can have a hazard associated with it. And then there is the risk of how much could I be exposed to that hazard. So if we just talk about the hazards associated with these, Amanda knows a lot about this as well and can speak to the different things that can go wrong. But let's talk about the things that are being done by these people inventing and developing these technologies. They're not using the native viruses. They're taking the best part of these viruses that can actually deliver genetic information into a patient's cell, but taking out all the bad stuff that would make that virus cause harm. Many of these viruses cannot replicate inside the host body. And that's typically what we think about viruses. They can get in the body in a potentially small amount, and now they get inside your cells and they replicate and make a lot more, and now you have a disease. The viruses are engineered to not be able to do that. That already reduces the potential hazard of these viruses versus what we just hear. Oh, this is a virus based on a lentivirus, and the lentivirus can cause AIDS. HIV is a type of lentivirus. And so people immediately react to that, but then they have to understand that these things have been modified to take out those virulence factors that actually cause the native disease. You can go through the list of these different products and how they've attempted to take the best of the virus and get rid of the worst. But there are things that can happen. One of these side effects that you would not think about is normally with a side effect of a drug, your body developing antibodies. So there are some types of these viruses that if you are exposed to enough of them, and we'll get into that back to the risks part, then your body can develop an immunity to them. And so if you ever needed that therapy in the future, you're not going to be eligible for that therapy because you've essentially become immune to that virus. That virus will not be successful in delivering the cargo into your cells because your immune system's going to fight against it.
Mark really hit on the most important point. We get so many questions in how do we handle these things, even at the bedside? Like, what are the risks to nurses? What are the risks to family members? Mark really hit on the most important points, and the antibody piece is the theoretical adverse event that I've seen most commonly. The other theoretical that could occur is some of these vectors do integrate into the host genome. For these integrating vectors, there's this theoretical that you could have unintended insertion where it's not supposed to be, and this could lead to mutations or cancer. We have seen some of this occur in very small numbers with some drugs. So that's the theoretical at this point for the gene therapies.
Are we concerned about these viruses penetrating intact skin? You talked about what happens when you put them in the body, but what about compounding pharmacy where someone might come in contact with the product? Are we worried about the viruses penetrating intact skin?
That kind of gets again to how do we classify these new biological drugs? What can we take for the infrastructure where we're already handling these other hazardous drugs? Talk about some of the other common hazardous drugs, chemotherapy agents, hormones. The greatest risk of these is dermal penetration, dermal permeation through your skin on a repeated basis of low amounts of these drugs. That's where the concerns are about occupational exposure to hazardous drugs. It's very unlikely, in the absence of a needle stick, inhalation, or ingestion, three things that we do great care to not have happen in a compounding pharmacy and through administration, these viruses do not go through your skin. They're too big. The likelihood that you're ever going to get enough of this virus in your body to have any of the negative effects that Amanda mentioned is extremely low. So that's where we talk about there's an inherent hazard here. We're talking about a lot of viral particles, but that the viral particles, when handled properly, are posing a much lower risk than we would see when we're compounding other typical hazardous drugs, whether in a sterile compounding environment or if we now get into non-sterile with hormones where it's like baking a cake and I've got the powder everywhere. And there's lots of opportunities for ingestion, inhalation, and dermal contact.
Okay, so they're hazardous, but they're not hazardous in the same way, and the risk is not as high as it is with chemo or hormones. Amanda, if hazards are not defined by the NIOSH list, how should these cell and gene therapy hazards be classified?
So essentially what we're seeing, we have this lack of guidance currently from NIOSH. So institutions are left to create their own policies. And what we're seeing is that we are relying on NIH risk level guidance and CDC biosafety level standards for the viral vectors, essentially borrowing from how these viruses and these vectors would be handled in a research lab setting. Currently approved vectors, they fall under BSL-1 or BSL-2 categories. How it's been explained to me and what I've been able to find in literature, even though BSL-1 would allow for you to prepare these agents on an open bench, in practice, pharmacies wouldn't do that. We are going to prepare these in a biosafety cabinet following BSL-2 standards. So you take this guidance and you apply this essentially in conjunction with USP 800 guidance, and that's how we shape these safe handling practices and the policies that we're implementing at institutions.
So, given what you just explained about whether or not these can be prepared in an open workbench or in a BSL-2 standard cabinet, at the end of the day, Mark, where would you say these cell and gene therapies should be compounded?
BSL, biological safety level, commonly used. We saw that in Outbreak. We're in BSL-4 and everybody's in bunny suits and all this kind of stuff. He's obviously at the lower end of the safety level concern, but we still have patients that we're going to administer these drugs. We're not just talking about reducing the risk of occupational exposure to these. We're ultimately delivering these to sick patients. We still need to do this in a very aseptic manner. When we talk about, well, where do I do that? I don't have a BSL-2 hood. Nobody's ever called it that before. I have a non-hazardous room and I have a hazardous drug room. And so help me fit the language that's coming from NIH and CDC and these manufacturers with what I actually have in my infrastructure now, unless I've got a facility that's willing to build me something that's just for doing this kind of therapy. But most folks right now, whether they're doing FDA approved or a lot of investigational products, they're going to have to make it fit in their current infrastructure. It looks like if you've got a hazardous drug room, you've got a CPEC, CPEC, that is the best place to do this. All the other measures that you would take to reduce occupational exposure, as well as to maintain an aseptic environment, are in place for that USP 800 room. Do you have to do all these in that room and fit the criteria? Probably not. But if you're going to move off of that, you got to think about a risk assessment of what am I sacrificing in terms of either occupational exposure or workflow or some other issue if I'm going to take that out of the 800 room.
So, Mark, you mentioned the engineering controls, and we know there's PPPE requirements for handling HDs per USP 800. But what about the deactivation, the decontamination, the cleaning and disinfection around compounding cell and gene therapies? Is it the same?
The concepts are the same, but how I'm going to achieve that and what I'm trying to do to it, in some respects, might be a little bit easier. If we come back and talk about what 800 tried to create this idea of, I want to break down the drugs, I want to deactivate chemotherapy and hormones, and I want to do this chemistry experiment on the deck of my PEC and my CPEC against these residues that might be there, that's hard to do. That's not obviously what these chemicals were designed for. They're designed to kill microbes. And even though we don't really talk about viruses being an issue with growing in CSPs, that's bacteria and fungi, we now have the opportunity to use a lot of these same disinfectants that have approval for killing viruses. There's opportunities here to still keep with the whole idea of cleaning and removing any residues, disinfecting other microbes that might get into my viral preparation. But again, if I have some escape of these viral particles, there are products out there that will work. Now, have they been tested against these genetically modified viruses? In most cases, they have not, because these viruses don't cause disease. So we've got IFUs coming from the manufacturers saying, just use a virucidal. Well, okay, now you got to get in and learn a little bit about viruses. There are viruses that have an envelope. These viruses, like SARS-CoV-2, like hepatitis, like HIV, they are easier to kill outside the human body on an inanimate surface. And things even like alcohol may very well be effective at being able to decontaminate or deactivate or kill those or inactivate those viruses. But other viruses like our AAV, adeno-associated virus, or adenovirus, so that's where a lot of the gene products are that people will be compounding, are non-enveloped. You'd say, oh, well, they got no envelope, must be easier to kill. No. If you've ever been on a cruise ship, think about norovirus. Norovirus, really hard to get rid of once it's outside the body on surfaces, and you do not want to get it inside your body. The products that you look on your disinfectant labels won't say AAV and may not even say adenovirus, but if they can kill norovirus or the different surrogates that they use for testing norovirus, like feline calcivirus, getting a lot of words in here, these are the kind of things that you look for that would be effective against these products. But then you've also got to go to the literature and understand, am I going to go get a product just for doing this, or can I make what I use today work in this case?
That makes a lot of sense. Amanda, what do you commonly see around this issue?
I love how Mark explained that, and I'm wondering how this lines up with what I'm seeing as a pharmacist and just reading the literature, because I'm not in the hospital setting compounding these things. But what I'm seeing in the literature is bleach, commonly a 1-to-10 dilution. I know there are some concerns with bleach damaging the cabinet. However, what I'm commonly seeing is you bleach and then you follow that with water and 70% isopropyl alcohol. I did see in a recent publication, Austin Wang was one of the authors. He's from Stanford. They proposed using Paradox, ready to use, as an alternative to the bleach. And that was the first time I had seen that. So I'm curious to hear from Mark if that's kind of in line with what we're seeing, if that would take care of things like the AAV vectors and the adenoviral vectors, because like you alluded to, that's most of what I'm seeing in the pharmacy pipeline are AAV vectors.
Okay, so we have to say what we know and what we don't know. These kind of products like bleach at the right concentration and wet contact time, anytime we're going to disinfect a population of microbes, we got to think about how long I have to leave that on the surface. And so the clock's ticking if I'm going to use bleach. But there are a lot of IFUs for these products that say use bleach. Now, how much data they have about what concentration of bleach for what period of time actually was effective at killing how many viruses, that's very defined when we talk about when these products are registered with the EPA as a virucidal. I don't know. I haven't seen the data coming out of these pharma manufacturers that backs up. Is that what I have to use, or is that just one thing that I could use? Because as you said, the bleach does cause some issues. Nobody at the pharmaceutical manufacturing or the labs was thinking about, oh, now they have to use sterile disinfectants in the CPEC. How am I going to dilute this bleach and keep it sterile if I could get it sterile to start with? So those are all the challenges there without over-promising that, oh, just use something that kills viruses and you'll be fine. You look at this as a hierarchy. If I'm using a product that can kill bacterial spores, nature's gobstopper, the thing that's the hardest form of life to kill, even more than a non-envelope virus, I should have high confidence that indeed, unless I've had a spill, that I could follow the normal practice of what I would do for a spore and follow that with those disinfectants.
Yeah, Mark, I like the way that you explained that. Specifically, a couple of questions we hear sometimes are, is it necessary to take that biological safety cabinet offline for an extended period of time? Do you need a dedicated biosafety cabinet? Is that necessary?
My understanding is no. The only commentary that I've seen that made me pause and think is, say you're using the same cabinet that you are using to prepare chemotherapy. These patients are obviously immunocompromised. If you do not properly decontaminate, you risk exposing immunocompromised patients to these vectors as well. So it's just taking that extra step to be cautious, to decontaminate. As far as taking these offline for any extended period of time or having to use a separate cabinet, no, it's not mandated.
That's good to know, Amanda. You guys have been great. This has been super helpful. But where can our listeners go for more information?
There are several publications by Jill Blind and her colleagues at Nationwide Children's. One of the most helpful in educating myself was Primer on Biosafety Handling, and that was published in the American Journal of Health System Pharmacy back in 2019. So we'll make sure to link that. And I really did enjoy that recent publication that I mentioned by Wang and colleagues from Stanford. And that was just published in the American Journal of Health System Pharmacy earlier this year. And Mark's webinar was absolutely phenomenal. I learned so much from it. That is probably one of my favorite resources. Highly suggest everyone check it out. I've been recommending it to all of my colleagues.
Thanks a lot, Amanda. I did not try to set you up to set me up, but you did. But thank you. Again, all I'm doing as a microbiologist trying to learn as much as I can from other experts. I'm not a virologist. I'm not a pharmacist, but I can take the science that's out there and try to explain it to folks that may not have this kind of background. I do that, but there's others as well. You talk about reading peer-reviewed journal articles, and the ones that you mentioned are great. They're not super complicated. Carrie Penzian at Michigan Medicine wrote a nice, easy-to-read article in Pharmacy Purchasing and Products in 2023 that talked about how do I assess and deal with these risks? How do I translate BSL levels to what I'm actually doing in a pharmacy? And then this is going on all the time. I just got all pinged about this survey from ASHP, the American Society for Healthcare Pharmacists, that they're looking for your opinion, the people listening to this podcast, about what are your questions about gene and viral vector therapies, and please respond to that. So just Google that, ASHP viral vector therapy, or go to the ASHP website, you'll find it. You can give folks this is what we need to know. These are the resources we need from an association like ASHP.
Yeah, we'll be sure to put links to all of the resources that you both just mentioned into the show notes, just to make it easier to find. Thank you both, Mark and Amanda. Thanks for joining us today and sharing your thoughts and insights. And it's been great chatting with you. Join us for more Verified RX podcasts. Subscribe today. Like us. Send us your comments. We'd love to hear from you. Verified RX is a prescription for success and is brought to you by the Vizient Center for Pharmacy Practice Excellence. Thanks for listening.